to start talking about catabolism, or the breakdown of biological fuel sources to make ATP. Those fuel sources can be all sorts of different things. They can be carbohydrates and lipids and nucleic acids and proteins, but they all kind of end up at the same place, this electron transport chain and oxidative phosphorylation. And so that's where we're going to start today. We're going to talk about this electron transport chain and how it can be used to power the synthesis of ATP through oxidative phosphorylation, a process we'll get more into in next lesson. So first of all, I want to give a little rationale as to why I'm taking this kind of bottom-up approach. Most of the time, if you learn about catabolism, you would start by kind of the top down. But with metabolism, there really isn't a top. And that's one of my goals this semester, is to try to convince you that there is no top. That yes, we can think about glucose as the feeder for this pathway. And glucose is the way that we often are getting our energy, but we can get it from a lot of different things. And even this simple thinking breakdown of glucose can actually, this pathway of like glycolysis and the citric acid cycle, these can actually be fed into and fed out of from these main ways if we think about these as isolated pathways. And so let's not think about them as isolated pathways. We'll look at the reactions in detail, as well as their regulation and their interconversion with other pathways. But at the end of the line, no matter what fuel source we start with, the way that we're getting this bulk of the ATP generated is through this electron transport chain and oxidative phosphorylation. And that process doesn't depend on where the original electrons that it's transporting came from. But first, so that we have a little bit of a perspective going into this, we're going to take that simpler perspective of glucose catabolism, or cellular respiration. Basically, we take glycolysis, where we break down glucose in order to get to pyruvate, and then we go through the citric acid cycle. Well, actually, first we have to oxidize pyruvate to get acetyl-CoA, which can enter the TCA, and then we can get into the electron transport chain. But none of the carbons of that glucose are actually getting into that chain. Instead, the only thing from that original glucose, or from any fuel source that's getting in, is the electrons. And those electrons are going to be carried from NADH or FADH2. And this is where the bulk of the ATP that's generated from all catabolic processes comes from. So if we look at glucose catabolism, we get 2 ATP from glycolysis and another 2 from the citric acid cycle directly, without having to go through this chain. And this is why the ATP that we get from glycolysis is enough for powering anaerobic respiration. And it's really the NADH2 that ends up being the issue there. But NADH2 that we generate and the FADH2 that we generate, both a little bit in glycolysis and then a little bit in the pyruvate oxidation and then a lot in the citric acid cycle, those are actually going to be powering the bulk of our synthesis of ATP. So the bulk of the NADH and FADH2 that's generated in cellular respiration is coming from the citric acid cycle. And well, the citric acid cycle, there's lots of ways to get there. And there's also a lot of other ways to generate NADH and FADH2, and we'll talk about just a few. So we'll talk later about how NADH actually can't get in or out of the mitochondria, and so the inner matrix, and so you have to actually shuttle in the electronic equivalents from the cytoplasm if you generate them there. But there's also a lot of reactions in the mitochondria that can generate directly there. All of these fuel sources, all different reactions, all sorts of different things. It comes down to the NADH, the FADH2, and the electrons that they bring. So let's start our in-depth look at catabolism with the electron transfer chain and oxidative phosphorylation. And then once we see how we can make ATP from the power of the electron transport chain, We'll go back and look in more detail about how we can actually generate the NADH in the first place and that FADH2. It's not just from glucose breakdown. And so we'll take an integrated approach to looking at metabolism. That's what to expect today, the electron transport chain. Next lesson, oxidative phosphorylation, or basically using the electron transport chain in order to generate ATP. Then coming up, lots more specifics on the various pathways. But for now, let's start at the bottom. Let's combine those principles that we've learned about thermodynamics, about redox, about membrane potentials. All of those things are going to come into play in this electron transport chain, which is part of the reason why I think it makes sense to start now at the bottom. Well, all these concepts are fresh in our minds, especially since, well, we can isolate that electron transport chain and just feed it NADH or FADH2 from anywhere. So let's do that. So oxidation and reduction, good old oil rig. Oxidation is loss of electrons and reduction is gain of electrons. If we pass electrons from something with a lower reduction potential, so something that doesn't want the electrons as much, to something with a higher reduction potential, something that wants the electrons more, that's going to be energetically favorable. So this is going to release energy. So if you hear energetically favorable, think energy is going to be released. So we want to transfer electrons from a lower reduction potential to a higher reduction potential, because remember, reduction, that oil rig, reduction is gain of electrons. If we pass electrons to something that wants it more, we're going to make the molecules happier. And if we make molecules happier, we get energy. We can then use this energy to drive processes like the production of ATP. But we need to controllably release this energy if we want to do that. If we look at, say, that NADH that we generated during the process of glycolysis and during the process of our citric acid cycle, our TCA, our Krebs cycle, whatever you want to call it, that NAD has a lower reduction potential than oxygen, much lower. This means that if we were to pass electrons directly from our NADH to oxygen, we'd get a big boost of energy. But we don't want just big booms going off in our cells. Instead, we want to controllably release that energy in a series of little steps. We can couple that energy to things like the movement of protons. And why would we want to do this? Well, in our mitochondria, we've got this situation where we have these different compartments. We can pump protons out of the inner part of the mitochondria, the matrix, into this intermembrane space. We can then build up a proton gradient, and not only is this going to be a chemical gradient, so because we have these physical entities, these different chemicals, we have this ion gradient, but we also, because these are charged, we're also going to have an electric gradient. So we're going to have an electrochemical gradient. We're going to be building up all of these protons in this inner membrane space. And then what's going to happen is those protons are going to flow through this ATP synthase like a dam that uses hydraulic energy. But in this case, we're using this proton motive force. Basically, we create this gradient that we're going to then use to drive the flow of the electrons through this pump that is then going to cause the pump to move, and it's going to cause ATP to be made and released, and then this can keep going and going. So we're coupling the passage of electrons to the passage of protons, which ultimately is going to drive the production of ATP. That's the big picture. By transferring these electrons in these little steps from one carrier to the next carrier, we're going to be pumping electrons because we're using the energy from those transfers from something with a lower reduction potential to something with a higher reduction potential. Energy released, energy used to create that gradient. That gradient is then going to drive the production of ATP. And now we just need to talk about all the nitty-gritty details of how this is all going to go down. First of all, let's talk about where this is all going down. When we talked about glycolysis, that was happening in our cytoplasm. When we talked about the citric acid cycle, and when we talk about the oxidative phosphorylation and our electron transport chain and all that stuff, Now we're going to be dealing with our mitochondria. So remember that our mitochondria, they came from basically an early cell swallowing a bacteria or similar prokaryote, and then therefore they have this kind of two membranes. So you've got this room within the room within your cell. This inner mitochondrial membrane is going to surround the inner room, this mitochondrial matrix, which is where our citric acid cycle was happening. The stuff that we're talking about today is mostly going to be taking place in this inner mitochondrial membrane, as well as in some of it in the matrix and some of it in this outer room, this intermembrane space. So you've got this outer mitochondrial membrane, you've got this inner mitochondrial membrane. Between them, you have this inner membrane space. We're going to be pumping protons out of the matrix into this intermembrane space. They'll then flow back into the matrix and generate the ATP on the matrix side, which can then get like swapped out so that it can flow out. This outer membrane is going to be much more permeable to things than this inner membrane. So this is going to make it so that we're going to be able to have things move more freely into this inner membrane space. But if you want things to go inside, well, now you're going to have special transporters, and this is going to come into play throughout. A quick note about mitochondria. So I've drawn them as kind of that simple cartoon, but mitochondria are far from simple. And they're not always a bean shape. So that was how people first saw it under a microscope. It looked like a bean, but it actually, mitochondria can look very different in different types of cells. Cells can also have different numbers of mitochondria. So our muscle cells, say, will have tons and tons and tons of mitochondria, whereas other cells that don't need as much energy won't have as many. Mitochondria can also do things like fuse together. And within a single mitochondria, you can have different amounts of kind of the folding of that inner membrane to give you these cristae. The more folding you have, kind of the more surface area you have to have those electron transport chain components embedded in them, so you can make more energy. In order to make all of those proteins that make up the respiratory chain, there you're going to have to make some of the proteins actually in the mitochondria themselves, and some of them are going to get brought in. So it's all this complicated stuff, but your mitochondria actually have their own genome, and it has their own ribosomes, and they actually make some of the proteins of the electron transport chain. We're not going to go into the details, but it is pretty cool. Going back to our big story, though, let's talk about how we're going to use the mitochondria and use that compartmentalization as a way to power the production of ATP. This is there's this chemiosmotic model. This is going to describe the coupling of ATP synthesis to the electrochemical proton gradient. So that's that gradient we were talking about before. We've got the chemical gradient, where we have basically the difference in proton concentrations is the difference in the concentration of a chemical. So we've got a chemical gradient. And we have an electrochemical gradient because we have these protons are positively charged, and if we separate them, well, then it's like a battery. So we're getting this electrical gradient. Put them together and we get an electrochemical gradient. When you move things down a gradient, that is going to be thermodynamically favorable. That's going to release energy, and that's going to be the energy that we're going to be using. So this energy that we create through the pumping of the protons is going to be used to generate our ATP. But we need to do that, create that gradient, and that gradient is going to be created through a series of redox handoffs. Remember that we want to move something with a lower reduction potential to something with a higher reduction potential. And therefore, we're going to need to have different molecules that can give and take electrons. And we want a variety of different molecules with different reduction potentials so that we can do things stepwise. And so that something will kind of always want it more until ultimately we get to oxygen, which wants it so, so bad. We saw a couple electron carriers when we talked about glycolysis and we talked about our citric acid cycle. These were going to be FAD and NAD. And then we also discussed how we could have like NADP plus which was going to be used primarily for anabolic things. But our FAD, so our flavin adenine dinucleotide, and our NAD, our nicotinamide adenine dinucleotide, we saw that in glycolysis, we created the reduced form of, we reduced NAD plus to give us NADH. And then in our citric acid cycle, we reduced FAD to give us FADH2. Keep this one in mind because it's going to come into play as actually part of our, that's going to be our kind of, the step where we create that is actually complex 2 of our electron transport chain. So stay tuned. But first of all, let's think about some of the similarities and differences between NAD and FAD. I know that I'm going to be kind of lazy about saying like NAD plus or NAD. Just go with it. What's the difference between them? Well, FAD, this is actually going to be tightly held in proteins. We call those flavoproteins. And whereas our NAD, this is going to be soluble, so it can go and travel around. The FAD is typically going to have a higher but variable reduction potential than our NAD. If it has a higher reduction potential, then basically, if we were to pass something to it, we would get more energy. And so we can use it to drive oxidation reactions that require more energy. Things like, say, making a double bond from 2 carbon single bonds. Whereas with the energy needed to, say, go from an alcohol to an aldehyde, for that, we could use NAD. But for going the harder reactions, we needed to use FAD. But if we use FAD, well, now we're at a higher reduction potential. And so we'll get less gain when we pass it off to the next thing in the line. The FAD reduction potential, though, is going to be variable because it's tightly held in the protein, and that's going to, different proteins will have different environments that are going to change the actual reduction potential. But it's typically going to be higher than your NAD. But those are far from the only electron carriers, and we're going to see a bunch of different electron carriers when we talk about the electron transport chain in this video. There are 5 main types of electron carrying molecules. So we talked about our NAD or NADP. We also have our flavoproteins. And so we talked about FAD, but there's also this a similar molecule, FMN, flavin mononucleotide, as opposed to flavin adenine dinucleotide. So very similar, but also a flavoprotein, so also very tightly held. And that is a molecule that is going to come into play in complex 1, as we'll see. Ubiquinone. So this is also known as coenzyme Q or Q. And this can move through the membrane. And then we have cytochromes, which are proteins with iron-containing heme groups, as well as iron-sulfur proteins. And so we'll talk a little bit more about these different ones. So remember when we talked about NAD and we talked about FAD, those were transporting pairs of electrons. Those were doing HYDRIDE transfers, remember? So we had that pair of electrons that was moving. When we talk about some of these metal carriers, when we talk about iron, when we talk about copper, these are going to take 1 electron at a time. And so we need a molecule that can go from 2 electrons, take 2 electrons and give 1 electron and that sort of thing. And it turns out that coenzyme Q is really great for this. So coenzyme Q is basically, it's got this little group where the action is going to be taking place, but then it also has this long hydrophobic tail. This long hydrophobic tail is going to allow it to hang out and move around the membrane. So that's going to be important in the CoQ's role of transporting electrons from one place to the other, that it's able to move through the membrane. You might have heard of like Q10 supplements. Basically, that 10 refers to there being 10 carbons in this chain, this R chain. But we're not going to focus on that chain right now. We're going to focus on where the actual oxidation and reduction are taking place. In ubiquinone, so our Q, this is going to be the fully oxidized form. Then if it takes an electron and a proton, well, now we get this stabilized free radical. So remember, a free radical is when you have a lone electron. So not a lone pair, but an actual lone electron unpaired. This is going to be our semiquinone radical, or our UB semiquinone. And so you'll see it drawn like this. What then happens is if it accepts another electron and proton, well, now you're going to get the fully reduced form, which we call ubiquinol, or QH2. So this is the fully reduced form. It required 2 electrons and 2 protons. And therefore, it's going to be able to give and take 2 electrons and 2 protons. And we can use it to both transfer protons as well as to transport electrons. It's ubiquinone, the name kind of suggests it's ubiquitous. It's going to act as a sort of universal carrier because of its role of being able to accept 1 or 2 electrons, and it's going to be able to take electrons from both our NADH as well as our FADH2 or our FMN. What about iron? So iron, we can have that like +3 state, that +2 state. Basically, it can give and take electrons, but it needs an escort. We need to keep it safe because we don't want things giving and taking electrons willy-nilly, or else we'll get unwanted radicals. So radical in the case of our ubiquinone was helpful because it's just this transitory thing. We'll see, however, how it can actually, if it escapes, it can cause oxidative stress, oxidative damage. When you have free radicals, those are often very reactive. We don't want metals just taking electrons or given electrons in an uncontrolled manner. And therefore, when we use atoms that, when we use metal ions that do give and take electrons a lot, things like iron, things like copper, we're going to use them in a tightly controlled manner where we have them held very tightly in things like proteins. And even within proteins, we'll have them held tightly, such as in prosthetic groups. So these tightly held molecules like heme groups. We saw heme in the case of hemoglobin, but it's also going to take to be here because, well, it's really good for holding onto iron. Also good for holding onto iron are like cysteines. So the sulfur, so this would be like an iron sulfur cluster, as well as you can have them coordinated in these things called like Reski proteins, where you have them coordinated by histidines. So various ways we can hold on to those metals and take advantage of their ability to give and take electrons. So we'll see these different molecules be used, things like our cytochromes, so our proteins that hold on to one of those heme groups and that holds on to the iron. We'll see our ubiquinone. We'll see oxygen ultimately, and we will see this NAD and the FAD. All of those are going to come into play in our electron transport chain. Now we need to look in a little more detail about how this chain is going to work. I think it's best to start with kind of getting an overview of what it all actually looks like. So there's this great animation from the Harvard X consortium thingamabob. And so why don't you go watch that, and then we'll discuss in detail. All right, so we've got these 4 complexes, and really complex 2, we can take that out and it wouldn't matter. It would matter in the terms of our final yield. But complex 2 is really just an alternative way to give electrons to Q. So remember Q, that's our ubiquinone. That's going to be the transferring, we can give electrons to it from things like NADH, which we do in this complex 1, or from things like FAD, or which we get indirectly from succinate, which is complex 2. There are also other ways we can get things to Q, and we'll discuss that later. But the role of complex 1 is to get those electrons to Q. The role of complex 3 is then to take those electrons from Q and pass them on to cytochrome C, this little protein with one of those heme molecules that's holding onto the iron that can hold onto the electrons. And then the cytochrome C is going to go, and it's going to go to complex 4, which is going to pass those electrons onto oxygen. And then this is all in these complex 1, complex 3, and complex 4. Here you're going to be pumping out protons, and the pumping out of protons, then we'll talk about how we can create ATP. But first, let's look in detail about how each of these complexes work. Don't get tied down by the nitty-gritty details, but take a look but start thinking about kind of the logic of the pathway and how we're passing electrons off. So in the case of complex 1, this is also called NADH ubiquinone oxidoreductase or NADH dehydrogenase. We're removing electrons from NADH, so we're oxidizing it, and we're reducing ubiquinone. So we're passing those electrons onto ubiquinone. But we're not doing that pass off directly. Instead, we're going to do it through a series of electron transporters. This starts with FMN. So remember, that was one of those. This is then like a flavoprotein. So FMN was our flavin mononucleotide. It was like our FAD, but slightly different. And it's going to be held in this complex 1 protein. It's then the electrons are going to be transferred from that through a series of our iron sulfur centers, ultimately to our coenzyme Q. Each of these pass-offs is going to be energetically favorable because we'll be going from something with a lower standard reduction potential to something with a higher standard reduction potential. We're going to couple the energy from those favorable transfers to the pumping of protons. So for each NADH that goes in, you're going to get 4 protons pumped in complex 1. And you're going to be reducing 1 Q molecule, giving it 2 electrons. So you're fully reducing it to its ubiquinol form. So we went from ubiquinone to ubiquinol. We now have this QH2, this ubiquinol, that we can use to pass energy on further. But first, before we go back to our story about the ubiquinol, we need to talk about another way we can form ubiquinol. And that's going to be our complex 2. So our complex 2, well, this just passes electrons to Q. And so this is succinate dehydrogenase. Yes, it's that succinate dehydrogenase. So it's that one from the tricarboxylic acid cycle, or Krebs cycle, or citric acid cycle. It was the step when we went from succinate to fumarate, and we actually made that FADH2. When we made the FADH2, this was actually happening in that inner mitochondrial membrane. So most of our citric acid cycle was happening inside of the matrix. But this step was actually happening in this inner mitochondrial membrane. This way, we have the electrons directly passed into the electron transport chain at this step. This is important because that FAD, remember, that's one of our flavins, that's going to be tightly held in a protein. So if we want to use this as an electron transfer, we need the molecule that's holding it, the enzyme that's holding it, to actually be where we need it. And so by having it embedded in this inner membrane, it's able then to directly transfer those electrons from succinate onto FAD and then onto ubiquinone, so our Q. So it's going to pass the electrons from succinate to FAD, then ultimately, through some iron sulfur centers to ubiquinone to give us ubiquinol. What's a little confusing is, yes, we're going to be transferring these electrons, but we're not actually going to be pumping any protons. So you might wonder, like, well, what's the point? Well, the point is to get the molecules to ubiquinone. So our ubiquitous carrier, that thing that's going to be able to transfer those electrons further down the line, we need the electrons to be there. So we're going to waste the energy from this in order to do that. Waste in the terms of we're not pumping out protons, but we're still doing something very useful. Why aren't we pumping out protons? Well, if we think back to the energy of our FAD versus the FADH2 versus the NADH, remember that our FAD had this higher reduction potential, which means that it's going to have less of a difference in reduction potential between it and Q than there would be between NAD and Q. And therefore, we're not going to get as much energy from this pass-off, but we still are going to get that ubiquinol. Now that we have that ubiquinol, let's go and let's take it further. Let's use it to transfer those electrons to cytochrome C. So cytochrome C, remember, that was going to be kind of like our relay protein. It's going to be this small little protein that can that can move around this inner membrane space. And it's going to be able to take those electrons from ubiquinone, ubiquinol, and transfer them to the fourth complex, which is where we're going to be transferring them to the oxygen. So complex 3 is going to also be called cytochrome BC1 complex, or ubiquinone cytochrome C oxidoreductase. So these long names, complex 3, a lot simpler to say. And it's going to basically pass electrons one at a time from our ubiquinol to cytochrome C. So remember, ubiquinol is fully reduced. It's got those 2 electrons it can transfer. Cytochrome C, it's going to only transport 1 electron. So cytochrome C, it's got that heme group that has the iron, and the iron is only going to be giving and taking 1 electron at a time. So we're going to need to just give cytochrome C 1 electron at a time. But we have 2 electrons in our Q, in our ubiquinol. And so we don't want to waste that second electron. Instead, we're going to go through some complex gymnastics in order to take ubiquinol and pass one of the electrons from it to the cytochrome C and kind of hold on to that other electron until we go to a second cycle. So there's this thing called the Q cycle. I don't need you to worry about the details, but I am just going to explain it so you have an idea about what's going on. Basically, you have these 2 binding sites for Q. You have this QH2, so our ubiquinol enters, and our ubiquinone enters, so our Q. So we have one that's going to be in this fully reduced state and one that's going to be in this fully oxidized state. What happens is that fully reduced one can now pass off those electrons. So it's going to pass 1 electron to cytochrome C. It doesn't pass it directly to cytochrome C. Instead, it goes through an iron sulfur center, but then it'll go to cytochrome C, and then cytochrome C can then move away and go take it off to complex 4. But we still have that second electron. That second electron, it's going to go through a series of heme molecule carriers, and it's going to actually reduce that ubiquinone that we had down here. So remember, we had ubiquinol up here, we had ubiquinone down here. The electron from ubiquinol, one of the electrons, kind of like the extra one that we couldn't directly pass to cytochrome C because cytochrome C would only take 1. We're going to take that electron and we're going to give it to this ubiquinone. This is going to generate that free radical, that semiquinone radical. Now what's going to happen is that this is going to leave us with another oxidized Q, and that Q can then move out and it can go and it can re-enter the pool. In the process of doing this transfer, we're also going to be transferring those 2 protons. So we're pumping out 2 protons in the first cycle. At the end of the first cycle, basically what's happened is that this free radical that we've generated, it actually moves back into position on this site. In order to reduce it fully, we're going to need to add 2 protons, and we're going to need to add another electron. We can give it another electron if we take another ubiquinol. So we take another of the fully reduced forms in. We take an electron from that and give it to this free radical to create QH2, to create our fully reduced ubiquinol. The second QH2, well, now it's going to give the other electron to cytochrome C. And so in this process, we basically passed off another electron to cytochrome C. And in the process of doing this, we're going to give off another 2 protons. And so we have the same cycle happening up here. But down here, this is kind of where we're going between having we're going from the fully oxidized to the semiquinone, and then to the fully reduced, our quinol, so our ubiquinol. So bottom line is we're going to be passing those 2 electrons from QH2 onto 2 cytochrome Cs and pumping out 4 protons. But it's not going to be as simple as just doing it straight like that. We're going to do it in a couple of cycles, but it is going to happen. Now you've got those cytochrome Cs that have those electrons. So at complex 4, now we're going to go and we're going to take those electrons from cytochrome C and pass them on to oxygen. Oxygen wants them really, really badly, remember? And so this is going to be highly energetically favorable. We're going to use it to drive the pumping of protons again, because we get that big boost, we get that protons pumped. And in the process, we're going to be making water. The numbers get a little weird here because when we think about reducing oxygen, well, oxygen, molecular oxygen hangs out as O2. And so we're actually going to take O2 and we're going to add 4 electrons to it. So we're going to need 4 cytochrome C molecules in order to do this. And we're going to create 2 H2Os. In the process, we're pumping out 4 protons. But if we were to think about it from the perspective of that initial NADH that was going into the line or something like that, well, then it would be the equivalent of 2 protons. So 2 protons per oxygen, but because it's molecular oxygen, it's O2, then it's going to be 4 protons overall. How this transfer is happening is it's going to be happening through several carriers. These include cytochromes A and A3, as well as copper ions. So you've got this kind of like iron and copper working together. So complicated transfers, but ultimately you're basically the same story over and over. We're passing electrons from something with a lower reduction potential to something with a higher reduction potential, generating energy, using that energy in this case to pump out protons and boost that proton gradient. Putting it all together, we can think of these 4 complexes. We have complex 1. We're going to take electrons from NADH, pass them off to Q. This is going to take our ubiquinone and give us ubiquinol in our Q pool. We can also give electrons to Q through succinate dehydrogenase or complex 2, which is the same succinate dehydrogenase that we had in that electron transport in our citric acid cycle. This is another way we can give electrons to Q. And it turns out that there are actually a lot of different ways to pass electrons to Q, including from things like fatty acid oxidation. So in fatty acid oxidation, we're going to need to do one of those reactions that requires the formation of a double bond and between 2 carbons. So we're going to use our FAD. And this is actually going to then contribute directly those electrons into this Q pool. So we talk about this kind of like general Q pool because it doesn't really matter where those electrons came from that are given to Q. Now you've got that reduced Q, you've got your ubiquinol. So wherever the source of the ubiquinol, now it can come and it can enter this complex 3. In the complex 3, those electrons from ubiquinol are going to be passed to cytochrome C. Cytochrome C, this is this little protein that can move in this inner membrane space. It's going to go and travel to our complex 4, which is where those electrons are then going to be passed off to oxygen, giving us water. In complexes 1, complex 3, and complex 4, we get enough of an energy boost from those electron pass-offs in order to pump protons out. But with complex 2, here we don't. There's not as much of an energy difference in passing those electrons from the FAD to Q than from NADH to Q. We don't have enough energy here in order to drive the pumping of protons, but we are able to contribute to this Q pool that we can then use to pump protons down the line. So if we think about electrons entering from NADH, well, for each NADH, we'll get to pump out 4 here. We'll pump out 4 here and we'll pump out 2 here. And so that would be the equivalent of 10 protons pumped out per NADH. And remember here, this is looking at it in terms of a single oxygen, so half an O2. So that's why you see this 2 protons as opposed to 4 protons pumped out, because we're talking about in terms of this kind of like half of the reaction happening. In the case of if we were to start with our FAD, if we were to start just by adding something to Q through one of these ways that doesn't directly go through complex 1, well, now we're only going to have these 6 protons pumped out. So when we come to accounting later on, we'll have to keep that in mind, that we're getting more energy. We're getting more protons pumped out if we enter at complex 1, but we can also enter at complex 2 or any other route to Q. And although we kind of look at these as these isolated things, actually these complexes often hang out in these kind of bigger groups called respiratomes. So the complexes themselves are often very complex and they have lots of different proteins. And then they hang out together in these respiratomes where they're kind of close together in the membrane, which makes sense because, well, you don't want those electrons flowing all over the place out of control. So to review, we can break down all sorts of different fuel sources, but through oxidative reactions in which we pass electrons from them onto other molecules. More specifically, we're passing them initially to NAD or FAD. In the process, then we can basically have these electron carriers, transfer those electrons all the way into the mitochondria. So to review, we can break down all sorts of different fuel sources through oxidative reactions in which we pass the electrons from the fuel sources onto electron carriers, our NAD and our FAD, and we can transfer the electrons from those through a series of electron carriers in the electron transport chain. By passing electrons from something with a lower standard reduction potential to something with a higher standard reduction potential, we're releasing energy. That energy is used to power the pumping of protons. The protons are pumped into this inner membrane space. This creates an electrochemical gradient. The great power of this electrochemical gradient is used to power the synthesis of ATP through a process that we'll talk about in next lesson. Through these series of carriers, we're able to generate this gradient. We have 3 places in which we pump protons, and then we also have carriers between them. NADH comes in at complex 1, or C1, and you pump out 4 protons. The electrons get passed from NADH through carriers onto Q, and then Q takes them onto complex 3, which pumps out another 4 protons, passes the electrons to cytochrome C, which then brings them to oxygen in complex 4, which pumps out another 2 protons. Note that we bypass complex 2 because complex 2, well, that's just another way to Q. Complex 2 takes electrons from FADH2, and this complex 2 is actually succinate dehydrogenase that we'll see come up in the tricarboxylic acid cycle, or TCA. So there's actually a lot of different ways to Q, and so we can actually take out complex 2, and this pathway would still work. We could take out complex 1, and it would still work. We just need a way to get those electrons to Q, which can get them to complex 3. If we come in at complex 2, however, we don't pump those initial 4 protons, and so we don't get as much ATP being made because we're not going to generate as much of a gradient. The gradient is going to be what we're going to get the power for this ATP synthase from, and that gradient is coming because you're passing the electrons from things with lower standard reduction potential to things with higher standard reduction potential. Now that we've seen how we can make this gradient, we're going to look and see how we can use this gradient.